very much. I want to appreciate you for the opportunity. Yes, my name is Delaren Dennis. I'm a clinical research assistant at National Hospital. I'm situated at Nigeria. Yes, and my research interests ranges from neurodegenerative, neuroinflammatory, and neurobehavioral changes caused by traumatic brain injury. I'm a member of the Neuroscience Society of Nigeria. Yes, and I'm open to further collaborations on neuroscience. Thank you very much.
Yeah.
I'm sure everyone can see my screen. I'll be talking on a proposed mechanism of reversal of established traumatic brain injury induced and anxiety-like behavior in rats using natural products and experimental study of honey. Yes, on to the introduction. We're going to be talking a bit about traumatic brain injury. Traumatic brain injury is injury that occurs due to the application of an external force to the head. Yes, it causes a wide spectrum of dysfunctions, and this is usually associated with certain pathological mechanisms such as excitotoxicity, inflammation, and more. Yes, based on severity, it is classified based on severity into mild, moderate, and severe traumatic brain injury. And it is also classified based on mode of impact. When I talk about mode of impact, I'm talking about, penetrating and closed head injury. So traumatic brain injury is classified into closed head injury. In closed head injury, there is no penetration of the skull, whereas in penetrating head injury, there is penetration of the skull. So yes, neurodegenerative disorders such as concussion, traumatic brain injury, and Alzheimer's disease have similar neuropathological conditions associated with neural cell death. Yes, traumatic brain injury is very common, especially in developed countries. As you can see from my slide, you will see that about 5.3 million people are living with traumatic brain injury related disabilities. And the most common disability that most traumatic brain injury patients live with is the anxiety disorder. It's an anxiety disorder. So hence, every traumatic brain injury patient is at risk of developing a post-traumatic brain injury anxiety disorder. Natural products with strong antioxidant properties can avert or reduce neurodegenerative disorders. Now we're going to be talking about the physical and neurobehavioral effects of traumatic brain injury on the brain. As you can see from my diagram, we have effective traumatic brain injury. So what does effective traumatic brain injury, how does it impact the physical aspect of a person and the behavioral aspect of a person? From here, you can see that it causes mood swings. Yes, it causes personality changes, anxiety, depression, sleep disturbances such as headache, changes in senses, seizures, loss of motor skills, decreased motion. It even affects the cognitive aspect of the person. You see that certain traumatic brain injury patients, they tend towards forgetfulness. Yes, they have trouble with orientation. Their fine motor skills are also damaged, problem with communication as well. It's hard for them to understand, it's hard for them to process, and all this can be overwhelming and tend to, head on to anxiety and depression. Now, looking onto the phases of traumatic brain injury, traumatic brain injury has three major phases. We have the primary injury, secondary injury, and neurobehavioral deficits. When we talk about the primary injury, this is injury that occurs upon impact. That is to say, upon once the external force gets in contact with the person in question, that injury that occurs instantly, that is known as the primary injury, whereas the secondary injury is injury that is more progressive, chronic. It occurs later on, few days after impact. And neurobehavioral deficit, these are just basically deficits occurring due to primary and secondary injury that are seen in the long run. We'll just take it to get a brief overview of the primary injury. What occurs upon impact? Upon impact in traumatic brain injury, there are various things that could occur. From my slide, you would see that the skull could get fractured. Yes, there could be cerebral laceration, that is to say, a tear in the brain. There could be cerebral contusion, that is a bruise in the brain, as well as vascular dissection, hemorrhage, bleeding in the brain, the blood-brain barrier is compromised. Diffuse axonal injury could also occur. Diffuse axonal injury is just injury to the axons at different areas of the brain. And this is actually very fatal because patients that normally have diffuse axonal injury have ended up with cases such as paralysis. Yes, so after this occurs, then the next thing, the body, the auto-regulated process, for healing kicks in, and we have the release of cytokines and chemokines, as well as damage associated to follow a damage associated molecular pattern. But I would just call this a repetitive degenerative process because at the end of it all ends up in cell death, which if not managed, continues, becomes a continual process. Whereas when we talk about the next phase of traumatic brain injury, that's the secondary injury, we're talking about cerebral flow dysregulations upon impact. Definitely the vessels are not spared. They are either dissected like we saw in primary injury, or they are autoregulated to begin what I know as vasospasms. Yes, when they are regulated to begin vasospasm, and this doesn't really work out most of the time. This then leads to increase in intracranial pressure and edema. Once intracranial pressure is increased and vasospasms occur, that is to say some blood vessels, contract, then the next step is hypoxic ischemia. Some parts of the brain will not be able to get oxygen. And once that part of the brain, oxygen is, in layman's terms, the food of the brain. Once that part is not able to get oxygen, the brain kicks in, its reflex process of protection and neuroinflammation. Neuroinflammation is actually supposed to be a protective process, but like I said, it is now more of a degenerative process that occurs in the brain. So from neuroinflammation, glutamate release, excitotoxicity takes place. Yes, excitotoxicity leads to mitochondrial dysfunction, which leads to generation of oxidative stress. At this stage, there is now an imbalance in the level of reactive oxygen species in the brain and the level of endogenous oxidants, that's antioxidants. That is to say, the brain can no longer cope. And this leads to impaired autophagy. The neurons, the cells, they are no longer able to repair themselves, and the long run ends up as axonal degeneration and neural death. This would look at this in detail later on, so hence I brushed over it quickly. Moving on to the neurobehavioral deficits, you can see that you would definitely have cognitive dysfunctions, anxiety disorders in the long run. Yes, this could lead to sensory disruptions as well, sleep disturbances, and more. Now, to just, expatiate a bit further on the phases of traumatic brain injury, as you can see from my diagram, they are, like we spoke, they are acute responses, and then as well, there are chronic responses. The acute responses, we've spoken about them, tissue damage down to apoptosis. And then I will just chronic responses, that is under the secondary phase of traumatic brain injury. As you can see from my, once there is damage, obviously the blood-brain barrier is damaged, there's release of excitatory glutamate. There is release of excitatory glutamate. Once this excitatory glutamate binds to certain receptors in the neurons, when I say certain receptors, I mean metabotropic receptors, AMD receptors. This leads to a massive influx of calcium, hence the polarization of the axon. This influx of calcium comes through a lot of channels, even through the sodium calcium channel in the cell. Calcium just floods into the cell, and this therefore leads to mitochondrial damage. It therefore leads to mitochondrial damage because the influx of calcium into the cell increases the level of reactive oxygen species, increases the level of phospholipases and other proteins. Phospholipases, I'm emphatic on that because phospholipases aid in the breakdown of the cell body of the mitochondrion in cell membrane of the mitochondrium, it's broken down. And once it's broken down, the mitochondrium begins to release apoptotic proteins such as caspases, calpains, cytochrome EC, and the likes. And ends up this ends up initiating the cell death process of that cell. And the thing is, once the cell death process of that cell is initiated, it happens in a necrotic way, in the sense that the cell bursts open. So that is to say, the glutamate, the calcium, the sodium, all these ions that started the initial cell death of the first cell, they are released and they go on to, cling onto other cells. Hence, it's now more of a cascade process. It's no longer, a process for protection. The aim has now been defeated. So that is the phases of traumatic brain injury. Now we're going to be looking at the neuropathology of TBI and anxiety disorder. When we look at the neuropathology of TBI and anxiety disorder, we see disturbed synaptic plasticity. We see enhanced neuroinflammation demonstrated by reactive astrocytes and elevated malondialdehyde. Another neuropathology, which is very common, is the loss of neurons and neurodegeneration. And as well, we noticed that malondialdehyde patients, traumatic brain injury patients, displayed increased pro-inflammatory cytokines, which are associated with mood anxiety. On that note, I would just like to give a breakdown of my most recent publication. This was honey-induced elevation of superoxide dismutase, declined lipid peroxidation, and how it attenuates neurodegeneration in weight drop model of traumatic brain injury. On this study, I was able to establish the neuropathological similarities of traumatic brain injury and other neurodegenerative disease, which is seen as neurodegeneration. Neurodegeneration was identified via the hematoxylin and eosin stain, as well as the Crecil fast violent stain. From the hematoxylin and eosin stain of my study, I noted a decrease in the neuropil. I noted, apoptosis. I noted also a decrease in the surrounding neuropil. That is to say, the axons too were not left out in the damage as well. And when we look at it biochemically, I also noted an elevated increase of superoxide dismutase. Superoxide dismutase is an antioxidant which helps to mop up the free radicals released during traumatic brain injury or any neurodegenerative condition. I noted an increase in the honey-induced model, and I noted a decline in lipid peroxidation. So therefore, by default, there was a decline in the malondialdehyde level in the honey-treated group. Upon this study, I actually came across various literature review that talked about the relationship of traumatic brain injury to anxiety-like disorders. There were a lot of research. Many studies had investigated the link between anxiety and traumatic brain injury. Neurodegeneration in Alzheimer's disease and traumatic brain injury causes disruption of the functioning of brain system involved in mood regulation. This could lead to manifestation symptoms of anxiety. Another research talked about the fact that TBI patients displayed increased pro-inflammatory cytokine levels, which are associated with mood anxiety. So many studies have been carried out to link, anxiety-like behavior with traumatic brain injury, drawing deductions from my previous research on the fact that honey, due to its high antioxidant content, such as flavonoids and phenols, was able to, reduce the level of reactive oxygen species and hence neurodegeneration in the brain. I thought to take it a step further and understand if honey as a natural product could also ameliorate the anxiety-like behavior found in traumatic brain injury. Hence the topic, honey on prevention of possible anxiety-like behavior in traumatic brain injury, an experimental study. The aim of this was to determine whether there is a correlation between neuroinflammation and post-traumatic brain injury anxiety in an animal model. In addition, I also wanted to examine the efficacy of honey treatment in reducing anxiety-like behaviors. So this is the methodology of the study. 20 adult male Wistar rats were carried out with an average weight of 120 grams. They were used in this study, and TBI was induced using the weight drop model. They were grouped into 4 main groups: the control group, the honey group, the TBI model group, group C, and the honey post-treated TBI model group D. The control group were given normal saline for 14 days. Yes, I actually chose 14 days because 14 days is the standard timeline for, repair to be seen. The honey group were given just honey for 14 days. The TBI model, we are not given anything. They were more of just, induced with TBI and left to rest. Whereas the honey post-treated TBI model were given after induction of the TBI, they were given 0.5 ml per kilogram of honey. The specific objectives carried out were just measurements of anxiety-like behavior using the elevated plus maze, serum enzyme activity for lipid polarization for MDA, and serum antioxidant enzymes were also measured, such as catalase and glutathione synthase. Well, this is the timeline. My study went on for about 17 days. From day 1 to 14, we had acclimatization of the rats. From day 0 to day 1, where the initial study began, there was induction of traumatic brain injury. From day 1 to day 2, it was established, and then honey was administered for 14 days to the last 2 groups, that is the honey group and the TBI model plus honey treated groups. Then from day 16 to day 17, yes, they were given their last dose and euthanasia was carried out. Upon euthanasia, the wet brain of the rats were measured for each group. And based on my slide here, you can see that there was a noted significant decline in the wet brain weight of the TBI model group, as opposed to the control group, as opposed to the honey group, and as opposed to the honey treated group. But upon analysis of the honey treated group, I noted there was actually a decrease when you compare the honey treated group with the control group and the honey group, but a slight increase when compared with the TBI model group. This decrease in the brain is attenuated to the massive neurodegeneration that occurred, which led to a decrease in, which led to death, and also a decrease in the surrounding neuropil of the neurons. So hence the decrease in the brain weight of the TBI model. So the slight increase that I noted in the TBI model plus honey treated groups, these just indicated that the antioxidant effects of honey were actually taking its toll on the animals. Yes, under the biochemical analysis of malondialdehyde and catalase, as well as glutathione synthase, there was an increased level of malondialdehyde noted in the TBI model group. Yes, there was an increased level and a decreased level of antioxidant enzymes. Whereas when you compare the honey treated group to the TBI group, as well as the control group, we noticed a slight decline in the antioxidant enzymes when compared to the control group. But when compared to the TBI group, we noticed a massive decline in the level of malondialdehyde, as well as an increase in the level of antioxidant enzymes. Now, the anxiety-like behavior of the rats were, studied using the elevated plus maze. Elevated plus maze is used to establish, to measure the anxiety-like behavior of various, most likely Wistar rats. Elevator plus maze has 4 arms. So rats, we know that rats, they are naturally explorative animals. Once you keep them somewhere, they tend to go about, but once they stay in one place in the closed arm of the elevated plus maze, they feel protected there. They don't move about. That is to show that rat is showing signs of anxiety. That particular behavior is an anxiety-like behavior in that rats. So based on my study, I was able to establish that the TBI model group, after the traumatic experience, most of them, once kept on the elevated plasmids, they moved quickly to the closed arm and didn't bother coming out. They just stayed there. So the conclusion of this study was that I noted an increased anxiety-like behavior in the TBI model, but this was decreased as compared to the honey treated group. The honey treated groups were able to walk out, go to the open arms, walk around the elevated plasmids, and that was really encouraging. Now, in summary, the summary of the study showed that anxiety-like behavior using the elevated plasmids showed the TBI model presented with anxiety, which was declined in the honey-treated groups. Results obtained showed that the TBI model presented with a statistically significant increase in the time spent in the closed arm as compared to a noted decrease in the TBI and honey-treated groups. The serum activity of malondialdehyde in the TBI model increased with a decline in catalase and glutathione synthase activities as compared with the control and honey-treated groups. Now, conclusions made from this study is noted that hence traumatic brain injury can induce post-traumatic anxiety by activating mechanisms of neuroinflammation, oxidative tissue damage that results in disruption of neuron circuits, presenting in anxiety-like behavior. And I recommend that honey, tested honey that contains high flavonoid and phenol content. Upon research, we noted that the Manuka honey was highly recommended to have high antioxidant content. I recommend that honey can be given as supplements to traumatic brain injury patients as a way to, prevent, ameliorate, and further protect against anxiety-like behavior in traumatic brain injury patients. Thank you very much. I would like to thank OSIS for this platform. I would like to thank my mentor, Dr. Memudu, for giving me the opportunity to share this research. And I would like to thank my association, Bingham University, for providing me with all I needed to bring this research to fulfillment. Here are my references. Thank you for listening.